Hello everyone. This presentation is about plant community and its method. This presentation is made by Vasawa Himanshu. So methods of studying plant community: floristic method, physiognomic method, and phyto-sociological method. So several methods are utilized from time to time. The investigation is mainly with respect to plants. All the methods are mainly with respect to plants. Number 1 is floristic method. Obsolete method. Flora is studied by listing various genera and species present in the community. Vegetation is described in terms of flora. It is a descriptive method. This gives no proper idea of the structure, composition, growth form, etc. of the community. Next is physiognomic method. Considering the physical characters, various species of community, they are studied chiefly in terms of their life forms, general structure, spread, etc. Ronquier's life form method became most popular. Records of different life forms, various species are recorded, distributed among different life forms like phanerophytes, chemophytes, cryptophytes, hemicryptophytes, and therophytes. Next one is biological or phytoclimatic spectrum. So biological spectrum, it represents percentage of total species in community. It represents the percentage age distribution of the species among the various life forms. Percentage value of each life form is calculated. From the biological spectrum is prepared. Percentage value, number of individual life forms multiplied by 100 and divided by total number of species. This is compared with normal biological spectrum. Example, you can see a chart in front of your computer screen. Next is phytosociological method. So phytosociology is a branch of science which deals with plant communities, their composition and development, and the relationships between the species within them. This method gives detailed information on the composition structure, species diversity, growth, trends of succession, other characteristics like sampling unit selected for the purpose of sampling. Mainly 3 forms of sampling units: area, line, and point. Area and lines are used. Definite area. Points is in thick with long grassy and shrubby belts. It becomes difficult to have a definite area. Number 1 is quadrate method. Sampling unit is a quadrate, maybe a square, rectangle, or a circle. Number 1 list quadrate. Species present in the area are simply listed. List count quadrate, along with listing numerical counts, are also made. Chart quadrate. Use of pantograph is done. Detail to scale growth and distribution in each species is recorded on a miniature graph paper. Pantograph is tracing device that help in drawing to identify, cover, and reduces the drawing to scale onto graph paper. Next is experiment permanent quadrate. The chart quadrate is left undisturbed and the area is studied for vegetational changes over long period of time. Study periodic changes in community. Useful in successional studies. Below point to be followed. Number 1, to determine size of the quadrate. Second is to determine minimum number of quadrates to be laid down. Third one is record of the species. Fourth, calculation of frequency, density, abundance. Second is transect method. Sampling unit is line. Sampling strip extended across a stand or several stands of vegetation. This is two types, line transect and belt transect. So line transect, it is a thin line used for grassland, and belt transect, belt for suitable land used for forest. The transect is divided into segments of suitable land, useful to analyze the vegetation changing in its composition through an ecotone. Each segment may be taken as a quadrate. Frequency density value of each species. So this is the point method. The movable points, they are inserted in a wooden frame, around 10. So pins laid down at random in the area. Plants hit by pins are recorded. Frequency percentage of each species is calculated. One hit of pin taken by a species is considered its occurrence. Thank you very much. Hello everyone.